Hello, this is Dr. Paul Cottrell, and I'm recording this on 5/13/2026 at approximately 10:24 a.m. We're going to go over the genome of the hantavirus and also a paper dealing with the differences between two different versions of the hantavirus that are human-to-human transmission. Now, what I want to start with here is this diagram that's showing the hantavirus and the schematic of a cross section of the actual virus. And then we're going to go over a little bit of the details of the genome. So the hantavirus is a negatively sensed RNA, single strand. And it's a segmented genome, very similar to influenza. It has a large segment, a medium segment, and a small segment. It also has a lipid bilayer and glycoproteins GNGC. The glycoprotein GNGC are the spikes. The attachment point primarily is the GN, and that attaches to the protocadherin 1 receptor. Now, the lipid bilayer actually comes from the cell surface. This is either through the Golgi apparatus or through the assembly at the cell surface. So the lipid bilayer is actually from the cell surface. The genome does not code for an envelope. Some viruses do, this one does not. Another feature that is important to mention is the ribonucleocapsid, the RNP. And what that basically is that you have a nucleoprotein that actually is protecting the RNA. Remember, this RNA is negatively sensed, it's segmented, and this ribonucleocapsid has a polymerase. So you have this nucleoprotein N protecting the genome, the RNA, and that segment has a polymerase. And each segment has that polymerase. So you have this RNP that's protecting the RNA. Now, what you have here is three segments, S, M, and L. And the size of the actual protein is, well, the way this is written, I wouldn't say protein, I would say actually nucleotides. But when you make the RNA, when the RNA is translated, it'll turn into a protein. And this S protein is going to code, the small segment is going to code for the N, which is the nucleoprotein. So the small segment codes for the nucleoprotein. The medium segment is coding for the GN and GC glycoprotein. And the L segment is coding for the actual viral transcriptase and the replicase enzyme. So it's going to transcribe and replicate. So this is really important in terms of replication and in terms of transcribing. Now, what I want to, so we have basically 1,290 base pairs. For the M, it's 3,616 base pairs and 6,456 base pairs. But I would prefer to just call that nucleotides. Now, this is another diagram that explains a little bit of the S segment, the M segment, and the L segment. So vRNA is the viral RNA, negatively sensed, and it has to be transcribed into through this transcriptase into the RNA, the messenger RNA for the N protein. So this S segment is approximately 1.6 to 2 kilobases, and the M segment is approximately 3.6 kilobases. So that's 1,000, so 3,600 approximately. Again, the viral RNA has to be transcribed into messenger RNA to code for this GP. This GP actually is the GNC and the GN, and it actually is cleaved. I have a video about the cleavage point, and the GN is the top part of the spike, the GC is the stalk. And then the L segment is 6,500 bases. And again, viral RNA, and that will code for the messenger RNA that will code for the replicase and the transcriptase. Now what I want to do is go over a paper that gives you some pause for thought. So let me switch over to that paper. This paper was published May 2021 and is dealing with the Andes virus strains. And there are two types. So this is dealing with what are the differential pathogenesis between the Andes virus strains CHI-7913 and Chile-97178. I'm sorry, Chile-9717869 in the Syrian hamsters. Now, again, the Syrian hamsters are from Fort Detrick, U.S. AMRID. Now, what is interesting in this paper is that Rocky Mountain Veterinarian Branch of the NIH, that's important to pay attention to. Also, Canada has been working on the Andes virus. So you have Winnipeg, and then you have Rocky Mountain. Now, let's dive into some of the findings of this paper, and then we're going to go over a little bit of the details. All right. Now, the hantavirus cardiopulmonary syndrome, HCPS, is a severe respiratory disease caused by orthohantaviruses in the Americas with a fatality rate as high as 35%. In South America, Andes orthohantavirus is a major cause of HCPS, particularly in Chile and Argentina, where thousands of cases have been reported since the virus was discovered. Two strains of Andes virus that are classically used for experimental studies of the virus are Chile-9717869, isolated from the natural reservoir, the long-tailed pygmy rice rat, and the CHI-7913, an isolate from a lethal human case of HCPS. An important animal model for studying pathogenesis of HCPS is the lethal Syrian golden hamster model of the Andes infection. In this model, Andes virus strain Chile-9717869 is uniformly lethal and has been used extensively for pathogenesis, vaccination, and therapeutic studies. Here we show that CHI-7913 strain, despite having high sequence similarities with Chile 9717869, does not cause lethal disease in Syrian hamsters. Now, it's important to mention here that Syrian hamsters are the disease model, animal disease model, to mimic what would happen in the human body. CHI-7913, while being able to infect hamsters and replicate to moderate levels, showed a reduced ability to replicate within the tissues compared with Chile 9717869. Now, let me just interject here really quick. Sometimes when something is coming from the natural reservoir and it goes into the human body, it sometimes starts to attenuate. And when it attenuates, it usually has mutations in the replicase, in the transcriptase. So this is really important. So we're talking about the L segment here. Okay, let me continue. Hamsters infected with the CHI-7913 had reduced expression of cytokine interleukin 4 and interleukin 6 and the gamma interferon compared with Chile 9717869 infected animals. So there was a reduced expression with CHI, which was the human lethal Andes virus that was isolated, compared to the natural reservoir of the Chile 9717869. So that's also interesting that there was a reduced expression of the cytokines. So there seemed to be a less of a immune response. All right. So with Chile 9717869 infected animals suggesting potentially limited immune-mediated pathology. These results demonstrate that certain Andes virus strains may not be lethal in the classical Syrian hamster model of infection. So something that could still kill the human isn't being captured in the hamster model. And further exploration into the differences between the lethal and non-lethal strains should provide an important insight into molecular determinants of pathogenic hantavirus infection. Now, as they dive into the differences, they're going to, through serial passage, recombinant engineering, and CRISPR, develop gain of function. That's a key point here. But also, just because it didn't produce death in the Syrian hamster model doesn't mean it won't produce death in the human model, in humans. Why? Because this actually CHI-7913 is an isolate from a lethal human case for the hantavirus cardiopulmonary syndrome. Okay, so keep that in mind. Importance: Andes orthohantavirus is a new world hantavirus that is a major cause of hantavirus cardiopulmonary syndrome in South America, particularly in Chile and Argentina. Andes virus is one of the few hantaviruses for which there is a reliable animal model, the Syrian hamster model, which recapitulates important aspects of the human disease. Here, we infected hamsters with a human isolate of the Andes virus CHI-7913 to assess its pathogenicity compared to the classical lethal Chile 9717869 strain. CHI-7913 had 22 amino acid differences from Chile 9717869. did not cause lethal disease in hamsters and showed reduced ability to replicate in vivo. Our data indicate potentially important molecular signatures for the pathogenesis of the Andes virus infection in hamsters and may lead to insights in what drives the pathogenesis of certain hantaviruses in humans. All right. So while they're doing this research, they're going to get gain of function. Now let's dive into what's the main finding here. Okay, in this chart, all right, we have the region, which is the S segment, the M segment, and the L segment. We already went over that. The S codes for the N protein. The M codes for the spike, the GN and the GC spike, and the L codes for the replicase and the transcriptase. Now we see now we have the nucleotide position of the genome, which is a negatively sensed RNA, then the sequence of the Chile version versus the Chi version, and what the amino acid change is in the position. So what we want to do is compare. Now we have 8 changes for the spike. Now, again, the spike is what attaches to the receptor. So that's important. Also, we know that certain mutations of the spike leads to chaperoning the spike to the cell surface for assembly versus through the Golgi apparatus. So, and we know that some of those positions are in the, I believe, the 1000 range. I think it was 1094 range. And I believe the other one was in the around the 350 range or so, maybe. Let me double check that. Let me just double check that really quick. I did a video on this for the increased shepherding to the cell surface. The modification was at the 537, I'm sorry, 532 position, and the 1094 position. So the 532, so approximately here, and the downstream of this at the 1090. See, the 1094 range. So approximately here and approximately here. Those mutations that I mentioned in that video, it's episode 7, they help with the shepherding to the cell surface. Okay, so that's an important feature. And when you shepherd to the cell surface and you're doing the assembly to the cell surface, you'll be able to bud more of the virons. You have more virons that are budding off the cell surface. Okay. Now, with the replicase and the transcriptase mutations, we have 15. All right. And that's important because the better it can replicate in the human body or in the animal model, more of this, more could be learned from that study. All right. Now, what can we glean from all this information? So we know that these mutations in Chi are less pathogenic in the Syrian hamster model, even though Chi and Chile can kill humans. All right. These are the Andes virus that can cause human to human transmission and have a high fatality rate. But Chi has a tendency to be less pathogenic in the Syrian hamster model. So to improve the chi for pathogenesis, since the mutations are mostly in the replicase and transcriptase through the L segment, you can have that L segment through genomic shift be incorporated into the Chile version or into the Chi version. So what I'm saying here is that in the laboratory, you can take this code here, the Chile code for the L segment that has this sequence, and replace it into the Chi, and you'll have a better replicase. And in that hamster model, most likely what will happen is you will see higher pathogenesis. So when we step back and take a look at everything that I have said about the hantavirus, everything, okay, to improve the pathogenesis of the current Andes strain that is floating around endemically, one can design in the laboratory or something that emerges naturally in the wild, where you have mutations I532K and S1094L for the GNGC mutations, which is in the M segment. That will help to chaperone to the cell surface to allow for the bilipid layer to be assembled with the genome, and have the L segment for the replicase code sequence here in the human to human isolates. So like the CHI-7913. So to make something more lethal, what you could do is you can take Chi-17913, put in that I532K and S1094L mutation in, and that for the M segment. That will help for the chaperoning to the cell surface. And then have this sequence here for these positions from the Chile strain into the Chi-7913 strain. Now, when you do that, you will be able to have a more lethal Andes virus. In addition, because of the hamster model, you can do serial passage through the hamster model to further enhance capability. And that's for in vivo. For in vitro, you can cell passage different tissues to improve the attachment of the G, N, and GC glycoproteins to the protocanherin 1 receptor. And the tissues that you want to focus on are the following. You would pattern, let's see here. The lower esophagus, the pharyngeal mucosa, the oral mucosa, and the tongue, the saliva gland, and the lung bronchial epithelium. And when you do that passage through those tissues, what will arise is a more functional spike protein or glycoprotein GN attachment. So, in summary, the hamster model obviously can be used to study pathogenesis of certain versions and potential therapies against it. But there is some limitations because some versions are less immune reactive, even though they are lethal in the human body. But sometimes when you have a virus that goes into the human body from a reservoir from, let's say, a rodent, it does attenuate and it's not as functional. And it kind of dies out. This is part of the reason why sometimes viruses will end up, there'll be an outbreak and then just dies out randomly because the replicase, because there's no proofreading, kind of breaks down. And this is what's happening here, kind of breaking down. And also because the R0 value to being able to contract it is so low that eventually the outbreak breaks down. Plus, for it to attach, right now, the current Andes virus needs to attach to a protocancerin 1, which is a common receptor on all cells, but it has more affinity to cells that are deeper in the lung. And this is the reason why it's very similar to the avian flu, where you got to breathe, you have to have very close exposure and breathe it in deeper to be able to have it attached to those lower lung receptors. It's similar. It's not exact. Now, in nature, you could have segments mixing and matching in cells that may have one version of hantavirus compared to another version of hantavirus, and that genomic shift could lead to further pathogenesis. So you can have an emergence through just mutations in the wild to do something very similar to what I'm saying to do in the laboratory. But the point here is that hantavirus can be supercharged, can be designed to be more lethal. Now, is it as bad as other respiratory viruses? No. But if you can train a mutation to attach to the protocancerin 1s to other tissues that are higher up in the respiratory tract, also in the esophagus, then you are increasing infectivity. So, in parting, because this is going to be the last video I do about this subject matter, unless something major happens, it's not the only way to make hantaviruses more lethal, but one way to do it is to make sure that the M segment codes for the 53, I'm sorry, the 532 and the 1094 mutation that I mentioned. So you have chaperoning to the cell surface. Also, you have the replicase code shown on the screen here, inserted into CHI-7913. And then that would be serial passage through the hamsters and the cell tissues to improve the attachment of the GN to the protocantherin 1 receptor. And when you have that, that's attaching to other tissues, not just deep in the lung, but higher up and also in the esophagus, then you're talking about higher pathogenesis and infectivity. So it's important to pay attention to this. Now, do we need to worry about this current outbreak that it'll end up being a pandemic? No. But in 2002, when SARS first started, there wasn't a big pandemic. There wasn't a pandemic. There was an outbreak. And the research on the coronaviruses were starting from Barrick's lab through NIH grants, going all the way back to the second quarter of 2001. So the SARS program platform has been in development for over 20 years. And the Hantavirus program reminds me, at the current state, reminds me of those early days of SARS. So because of artificial intelligence and our understanding and capability of molecular biology and biotech, the development of these are much faster. So it's not going to take 20 years to go from the issue we had with SARS-1 to SARS-2 for the Andes current outbreak to some future outbreak. I suspect that we will see in the future something much worse that will be more problematic and be classified as either a category 2 or a category 1 bioweapon. Right now, it's a category 3 or category C, depending on how you want to classify it. So that's where we're at. Please help support the work that I do. All these videos are for free. And I think I give you a unique perspective on how this stuff is being made. Now, you can share the links, ask your social media to follow me. Make sure you click the like button and make sure that you subscribe to all the channels I have. I have YouTube, Brighteon, Bitchute, and Rumble. Just in case one of them gets pulled down, it's important that you get the information. This is the reason why I have so many different channels. So please subscribe to all the channels. In addition, follow me on X and GETTR. Those are other modalities to find the work that I do. And if you like the content and you want to help support, you can donate on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee. You can be a paid member to my Patreon channel and Substack channel. I do all these videos for free, so please help support the work that I do. I don't put anything behind a paywall. Because if I'm going to say something, I think everybody should know. I don't like when people put stuff behind paywalls. So, but I do need support to be able to do all these videos for free. So please help support and take a look at the great products that I offer. If you pay attention to my videos, you'll understand why. So please go to my website, the-studio-reykjavik.com. Thank you for listening. Hopefully you've learned something through this series on the Hantavirus. My understanding is I'm the only one telling you how this is being done, and I give you some important insights. And not only am I giving you the important insights, I'm actually showing you on the screen what the mutations need to be to make it more lethal. So thank you for listening, and have a nice day.